Okay, so you just have to be careful now when looking at the structures of our polysaccharides. For chitin, it is a derivative of our glucose, which means instead of the OH you can see in the normal glucose units, we have the substituted OH here. Instead of OH, you have the amine, but the amine is further reacted to form C double bond OH3 here. That is our chitin. Okay, another kind of polysaccharide is you have the dextran. So the dextran are usually bacterial and yeast polysaccharides. Okay, so that's their source. You have from bacteria and yeast polysaccharides. It's made up of the alpha-1,4. You have here alpha, it's down, and carbon 6, not 4. Carbon 6, so alpha-1,6 glycosidic bond, and for a glucose, down, down, up, down, down, up, down. This is up ha. Mura lang siya down kay nakatuwad ni, naka-incline siya. This is up. Ngayon mo na siyang ipa-straight. This is actually up. This is actually glucose. Down, up, down. This is our Dextran. This is our glucose and glucose that we're using here. The alpha-1,6 glycosidic bond. Alpha. Okay. You have to remember alpha, beta. Alpha ba or beta ba ang gamit ng glucose, and then type glycosidic bond, the 1,6 glycosidic bond, and of course GLC, that's glucose. I repeat here. So it's made up of alpha-1,6 glycosidic bond between glucose units, no? And there is a branching for every, all have alpha-1,3 branches. So pag abot nimo sa carbon 1, 2, 3, na branching na for every green part, carbon 3. No, so this is the difference between dextran and the other branched polysaccharides like your amylopectin, no, and glycogen. No, so remember the branching is on carbon alpha-1,6 glycosidic bond. That's their branching. But here, and of course, that one utilizes for our other polysaccharides here, amylose, amylopectin, glycogen, they form an alpha-1 for glycosidic bond. In this case, and they branch lang siya sa alpha-1,6. In this case, the dextran wala siya alpha-1,4 glycosidic bond, but the main straight chain niya is alpha-1,6 glycosidic bond, and there is a branching between alpha-1,4 glycosidic bond and sometimes also alpha-1,2 or alpha-1,4. So dili siya specific sa dextran. So pwede siya mo branch diri carbon 2, alpha-1,2, pwede siya mo branch diri ang carbon 3. Alpha-1,6 glycosidic bond. And alpha-1,4, so 1, 2, 3, 4. So carbon 2, 3, 4, pwede siya mag-branch. So very specific sa dextran. So where can you find dextran? So kanang mga dental plaques na to, it's formed by bacteria growing the surface of teeth. Kanang yung mga plaque, tanawan na yung mga ngipon kung na ba may mga plaque, no? Because it's made up of bacteria and yeast polysaccharides. And kanang plaque na ito, makita sa itong mga ngipon or somewhere else, no? Kanang sa mga bungkod, no? Are rich in dextran. So synthetic dextrans are chemically cross-links, or actually synthetic dextrans, other than what you can find sa kanang may mga plaques sa inyong mga ngipon. Synthetic dextrans are chemically cross-links to form insoluble material used in size exclusion chromatography. So this one, sometimes dextrans used in separation science, in organic chemistry, to separate different compounds. But normally, or in natural setting, you can find your dextrans, no, from the bacterial growth on the surface. And an example is kanang dental plaque, but you can also find dextrans in some other surfaces, no. that is formed by bacteria and yeasts. That's a polysaccharide. It's a highly branched. The other kind of polysaccharide is you have the hyaluronic acid. The hyaluronic acid is made up of glucuronic acid, so it's still a glucose, no? But modified glucose or derivatized glucose. Inside you have your up, down, carbon 5, carbon 6 forming a bond with carbon 1. It needs to rotate here. And carbon 6 instead of CH2OH na natay double bond OOH, no, the glucuronic acid you have here. And then the second monosaccharide is N-acetylglucosamine. The N-acetylglucosamine, okay, you have here NH. Muna siya ang atong monomeric unit ni chitin. No? You have NH double bond OCH3. Okay, so you have here carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, carbon 6, H2OH. No? So it's formed by the beta glucuronic acid and apmani. So it's also can either be, you have your beta also, glucosamine, and acetylglucosamine here. So they form a beta 1,4 glycosidic bond between your glucuronic acid and acetylglucosamine. And this is the repeating unit of hyaluronic acid. So asa na ito makita sa hyaluronic acid? If you look at na kaninyong mga pambutang sa inyong mga now, the creams and all others, no, mga gipang advertised karon, they're usually made up of hyaluronic acid. So hyaluronic acid, you can find that ingredients in lotions, moisturizers, and cosmetics. So sa iyang kuan, makapakuan na siya, makapahami sa now actually. Now, why is that? So hyaluronic acid is the simplest acid polysaccharide present in connective tissues. Kaya mga connective tissues na to. No, it's you have hyaluronic acid in there. It has a molecular weight between 105 and 10 grams per mole. And our repeating units here, you have 300 to 100,000 repeating units, depending kung asang organ which it occurs. Okay? Basta-basta na yung mga connecting tissue na to, kanang i-flip mo, no, sa itong kuan, itong mga organs. There's a connective tissue, connects one tissue to another. Hyaluronic acid is always present. So, yung repeating units depende kung unsang organ. And so, it's most abundant in embryonic tissues and in specialized connective tissues such as synovial fluid, the lubricant of joints in the body, and the vitreous of the eye, where it provides a clear elastic gel that holds the retina in its position. So, you can find your hyaluronic acids in there, even in embryonic tissues. And I've mentioned that you can find hyaluronic acid sa inyong mga cosmetics, sa inyong moisturizers, lotions, no? And you can actually, if you have hyaluronic, let me see, yeah, kanang inyong itong gel bitaw, usually you can find sa Watsons, katong orange, no, na siya hyaluronic acids. Tanawan ninyo, whenever you look at the ingredients, tanawan ninyo yung hyaluronic acid kung na-particated. So before, no, there's naka-discover hyaluronic acid, and the trend nowadays in cosmetics and personal care products, ginabutangan nila og hyaluronic acid kay makapahamis daw, no, it slows down aging, no, it stretch your skin cells para mahamis siya. So hyaluronic acid, again, as I mentioned, you have the glucuronic acid and N-acetylglucosamine joined by beta 1,3 glycosidic bond, which is in turn for the glucuronic acid and N-acetylglucosamine. So for glucosamine, you have your beta 1,4 glycosidic bond. So beta 1,3, kita na to, beta 1,3 glycosidic bond. And then pag diri na ta, you have your alpha 1,4 na dione, like alpha beta. This is up, up. So you can see here, this is a beta. So kung sumpayan mo niya, kanya i-repeat mo na kay saan niya. So this one is a beta, this is a beta also up. And pag between glucuronic acid and acetylamine, it's beta 1,3 glycosidic bond. Pero kung i-repeat nato ni siya, dili nato isumpay. So kani ang sumpay niya is kani si glucuronic acid kay naman ni. So kung kana siya sa kung siya masumpay, so it is formed by alpha up na ni beta 1,4 kani 4 glycosidic bond. Okay, so there is two kinds of bond that you can find in hyaluronic acid. The beta 1,3 glycosidic bond between glucuronic acid and N-acetylglucosamine. And pag ma-repeat na to siya, you can also find the beta 1,4 glycosidic bond between glucosamine and glucuronic acid. Kung nag-una si glucosamine at si glucuronic acid, nag-una si glucosamine, kani, and then kani siya, no? You have the beta 1,4 glycosidic bond. Okay, so another kind of polysaccharide, you have the heparin. So, someone is the heparin. So you can see here in the structure, mura siya N-acetylglucosamine, but modified siya further. Kaya na siya sulfate group here. So down, up, down. Si glucose, no? Glucose yung structure. This is down. Here, carbon 1. So it's like an alpha glucose, pero modified nga glucose. So unsa man si modified glucose? Instead of OH here, na siya NH, C double bond OCH3. So you call this one N-acetylglucosamine. But napa siya ni. So N-acetylglucosamine napagig siya 6 sulfate. No, so further modified kaysa kang chitin and it's momentong natuan kani ang hyaluronic. Si chitin og si chitin o kaning si chitin lang may N-acetylglucosamine. Okay, what else? So you can have carbon 1, 2, 3, 4, and then you have your COO. COH or COO minus, and somebody, this is your glucuronic acid. Or glucuronic acid. You have your glucuronic acid. You have here. So N-acetylglucosamine 6 sulfate or 6 sulfate in acetylglucosamine you have here, and then you have your glucuronic acid, and then napilaing diri. So instead of NH kani, you have your sulfate, N-sulfate, no? Derivative of your glucose. And then carbon 2, carbon 3, you have also the sulfate. So what you can see with heparin is the kind of sulfate, no? Carbon 2 sulfate, NHSO3 sulfate. No? So variable siya. So heparin is a heterogeneous mixture. Heteros siya. There's too many kinds of monosaccharide that you can see. You can find this glucuronic acid, then you can find a derivative of this instead of kani, NHSO3 sulfate. And then another na pod, no? Instead of NHSO3, napagay kay OSC3, and then napagay COO. So another na pod, you have your NHSO3 and napagay SO3. So pila siya kabuok. You have 1, 2, 3, 4, 5 types of monosaccharides. Nag-repeat. Nabalik-balik. So different manner. So heparin is a heterogeneous mixture of variably sulfonated polysaccharide. But what you can see normally is for most of the structure na siya sulfate, SO4 to SO4, okay, minus added sa yung structure. So heparin is a heterogeneous mixture, as I mentioned. It's sulfonated, and the molecular weight is around 600 to 300,000 grams per mole. So where can you find that? Acidic polysaccharide. This acidic polysaccharide, acidic man siya, no? Kaning SO3, this is the dissociated form of the sulfonic acid, actually. The acidic polysaccharide is synthesized and stored in mast cells that are part of the immune system. Okay, natin makita that occur in several types of tissues, various parts of tissues present sa liver, lungs, or even sa gut. So, unsay function ni heparin? Heparin is best known and fully understood as an anticoagulant. Okay, and because of that, actually, most of the kaning mga dressing, no? Kanang ipangbutan ka ng grabe nga sa mud, inabutangan ng ilang heparin. Anong heparin kay because it has an anti-coagulant activity. So instead nga magsigig dugo-dugo na kung nasamad na iyang i-cause of coagulation, dili mo ipaklat niya ang blood. No, manang gigamit siya as sa mga dressings. So heparin, so it has anticoagulant activity and it binds strongly to antithrombin 3. It's a plasma protein involved in terminating the clotting process. So dali raman siya mo form sa antithrombin na present in blood or plasma protein involved in terminating the clotting process. Okay, so a heparin preparation with good anticoagulant activity contains a minimum of 8 repeating units. Can easily have 1, 2, 3, 4, 5. But usually they have an 8 repeating units. And the larger the molecule, the greater its anticoagulant activity. So because of its anticoagulant activity, it is widely used in medicine. And I have mentioned that one of those is katong sa mga dressing wounds, no? Addressing ipangkuan sa wounds. So let's summarize our polysaccharides that we have discussed so far. So we have 1, 2, 3, 4, 5, 6, 7. Wala ni appeal ang peptide glycan agars. Okay, so far we have discussed about the starch. It is the energy storage in plants. You have also discussed the glycogen, which is an energy storage in bacteria and animal cells. So, anti-glycogen can be stored. Glucose na to is converted as glycogen. Okay. So for starch, there are two kinds. You have your amylose and amylopectin, and they are both polysaccharide, which means made up only of one type of monosaccharide. In this case, you have your glucose. So parehas here, the linkage is alpha-1 for glycosidic bond between glucose, and it is linear. So the size is about 50 to 500,000 monosaccharide units sa amylose. Si amylopectin, the difference ng amylose is that aside sa alpha-1 for glycosidic bond, na linear, napod siya branching. No? So branching niya, it utilizes the alpha-1,6 glycosidic bond every 24 to 30 residues, or 25 to 30 residues. Okay, another kind, you have the gluco glycogen. It's a polysaccharide again. You utilize glucose, usually glucose. It's similar to the amylose and amylopectin. You utilize the alpha-1 for glycosidic bond. And sa branching, your branch siya, pareha siya kang amylopectin. Na siya branching that occurs with alpha-1,6 glycosidic bond. But the branching, yung difference ng glycogen kang amylopectin, kani, nag-branch siya every 24 nga residue or 30 residue, 24 to 30 residue. Kani branch siya every 8 to 12 residues. Mubo ang iyahang sa branching. Okay? So, the size is up to 50,000 monosaccharide. Okay? It's an energy storage in bacteria and animals. So, mas taas o monosaccharide number C, amylopectin. Okay? Mas taas na siya mo branch. 24 to 30. Glucose man. So cellulose is glucose gyapon na itong repeating unit, homo gyapon siya, but we don't utilize an alpha glucose. We utilizes a beta glucose. So beta glucose, the glycosidic bond is between the beta 1,4. The first glucose is a beta 1,4 glycosidic bond. Of course, expect the second glucose to be also beta. The continuous man siya, beta 1,4 glycosidic bond. So the number of monosaccharides is up to 15,000 monosaccharides, and where can you find cellulose? Of course, sa mga cell walls, and it provides rigidity actually sa mga cell walls, sa mga parks, sa mga any plant that you can find, sa gawas that protects the plants, no? It's made up of cellulose. The next one is you have your chitin. Chitin is a homo, and it's made up of glucosamine. No, it's a derivative of glucose, GLCN. So glucose yung a derivative but N-acetyl. So na N-acetyl on carbon 2. Okay, and that is your N-acetyl glucosamine. And the link, we're utilizing here the kind of N-glucosamine which is beta. So glucose siya nga beta but derivative sa beta glucose nga na siya N-acetyl glucose. Unsa lang N-acetyl katabi tawag instead of OH na siya NH C double bond OCH3. Okay, and then so the type of glycosidic bond that's being utilized here is beta 1,4 glycosidic bond. So magbalik-balik ko na siya. Atong carbon 1 is beta and the fourth carbon of the glucosamine and glucosamine you have here. So this your chitin, you can find that in insects, spiders, crustaceans, which gives rigidity to its exoskeleton structures. And then next you have your dextran. So dextran is again made up of glucose. It's still a HOMO, but it utilizes the alpha 1,6 glycosidic bond between glucose. So you have your alpha, kani ang gamit niya dito sa amylopectin rather and glycogen sa branching, no? Siya dito magamit. But sa dextran mo ni iyang main straight chain glycosidic bond, alpha 1,6 glycosidic bond. And then mo branch siya for every alpha 1,3 glycosidic carbon 1 and carbon 3 between the monomers and utilizes the alpha 1,3 glycosidic bond. So where you can find this, it's in bacteria and extracellular adhesives. It's also the function is structural. And then we have the hyaluronic acid, no, you have mentioned that one. The hyaluronic acid, you have the glucose and the N-glucosamine. This is hyaluron because we're utilizing a glycosaminoglycan or the glucose and then the N-glucosamine you have here. You have your glucuronic acid rather and your N-acetylglucosamine. GLCA, so glucuronic acid and GLCNAC. So this is your glucose but derivatized with N-acetyl. We call this one with N-acetylglucosamine. Okay, and you have it forms a bond between glucose and glucosamine to be beta 1,3 glycosidic bond, but you just have to be careful na apa siya aside sa beta 1,3 glycosidic bond na apa siya 1,4 beta 1,4 glycosidic bond. So beta 1,3 glycosidic bond, then apa siya beta 1,4 glycosidic bond between this glucosamine and glucuronic acid. Okay, so where can you find this one? It's structural. You can find this in connective tissues and in lubrication, in viscosity and lubrication insurance. Okay, so it's very viscous and it's used for lubrication insurance. Okay, and the two that we didn't mention, there is another one. You have the peptidoglycan. Peptidoglycan is it's a peptide. There's a peptide bond actually being used, and then you have your find this one in bacteria that gives rigidity and strength to cell envelope. So you have the beta 1,4 glucosamine. Okay, I will look at the structure of this. And you have the agarose, no? So here what you can find is a hetero, there's a peptide attached, this is where the peptide, and you have your glucosamine that you can find in your hyaluronic acid and the glucosamine that you can find in chitin. And then you have the agarose, no? You can find that in algae, so in its cell wall. You have the D-galactose and the 3,6-anhydro L-galactose. So it utilizes the stereoisomer of galactose, which is the L-galactose, and anhydro, so which means there is no hydroxy bond bonded to your L-galactose here. Okay. We look at the, wala dito ang agarose, no? So look at the other. And okay, this is Ramila 15. I just have to read on this chemical connections called in the life-saving carbohydrates, no? Chitosan. Chitosan, you have, this is the, if the N-acetyl group is removed, remember you have your NH2 for your chitin, NH2, CH3, you have there. And once it's being removed, no, you have here the chitosan. Actually, you call it one is a chitosan. Pagkatong N-acetyl, C double bond, NH, C double bond, OCH3, pag na-remove to si C double bond, OCH3, katong dili, from your chitin, no, katong chitin structure, natay NHC double bond, OCH3. Kung ma-remove to siya, you have here, you get actually your chitosan. So chitosan differs from cellulose in that it has the amino group. You have your NH2, but in water, si NH2, protonated siya, may mo siyang NH3+. Okay, so may difference ni cellulose and chitosan. This is the chitosan. Chitosan also is a derivative of cellulose and chitin. Si chitin na siya NH2, so NH2, CH3. So asa man siya ma-derive natin si chitosan. This is the structure of chitosan. Ma-derive natin na siya katong natay chitin, unya atong i-remove si acetyl group, then you can have your chitosan. So asa natin makita si chitosan? Actually, akong mentioned because you can also find our chitosan, that's another kind of polysaccharide sa mga dressing food nato. So aside from heparin, but akong mention ka ganina, no? Aside from the heparin, okay, you can also find your chitosan, no, in in the dressing rather. Okay, so why is that? So because it is protonated here, it is charged, no, it's possible for the opposite charges, no, to interact with the red blood cell that is a net negative charge. So pag you can see in the structure here, it's positive. And if you look at yung mga samad, no, na maging blood, no, the blood has a red blood cells, contains red blood cells, and it has a net negative charge. So pag interact si chitosan, kaning structure o katong si blood, no, so what happens is that na interaction, which is ionic, and because of that, it produces a blood clot and stops the bleeding rather. Okay, so you have here the chitosan being utilized as a source for wound dressing. Okay, so maybe I can include this. And I will include this one sa inyong, or you can just read in the book, no, and read in this. So basically, we have already covered, what we're supposed to cover for the polysaccharides, part of your carbohydrates. So we're already good, and just if you just want to have an additional reading, you can read on that. So there's just two kinds of polysaccharide, which is more than sa inyong ginagamit, no? Itong encounter, you have the peptidoglycan and the agarose that we haven't discussed. But agarose, you have if you're in biology and you use, you do want to isolate, for example, microorganisms, you use the agar, and it will be one of the source for your carbohydrates, polysaccharides, and the agarose. Okay. But you haven't shown the structure, so anyway, this two will not come out. We just know that there's another kind of polymers that we can see. Okay, so that ends our discussion for today at the end of chapter, and you can look into problem set. The problem set, we have first problem set. Now there are 6 people who didn't submit the due date na siya. And then you have problem set number 2. Which is about carbohydrates. So wala na ako nag-quiz, no? But here, problem set number 2. So which of the following compounds are monosaccharides and which are L-monosaccharides? Define the term anomeric carbon. Which carbon is anomeric carbon in glucose and fructose? Of course, you have to show in the structure. Define pyranose and pyranose. Explain the conventions for using the alpha beta and beta disaggregate configuration of the cyclic forms of monosaccharides. Are alpha D-glucose and beta D-glucose anomers? Explain. Are they enantiomers? Explain. Convert each of the following chair conformations to an open chain form and to a Fischer projection. You have here, this is the chair conformation in cyclic form. And in the mutation process, you can convert this into straight chain and straight chain to this one, from the Fischer to this projection. You can have, so from this, you can name on what kind of monosaccharide this one. It's not easy to see with this. You have to look, because the list that we had is in the Fischer form. Okay, and this one is in the cyclic form. Again, when you have your cyclic form, you can obtain this when the straight chain Fischer projection undergoes mutation. Mutation ito form siya cyclic form at equilibrium in solution. In solution, of course, you know that majority of the monosaccharides exist in the pyranose or furanose form, in cyclic form. So you have your D-fructose. It can be reduced by sodium borohydride, which gives 2-aldol, one of which is D-sorbitol, named under the structure. Okay, you have one of the reactions that we have discussed. Name 3 important disaccharides from which monosaccharides is each derived. What does it mean to describe glycosidic bond as beta-1,4, alpha-1,6? So here is a trehalose. It's found in young mushrooms and is the chief carbohydrate in the blood of certain insects. So name the 2 monosaccharide units. Describe the glycosidic bond. I think we have already discussed that one before, but anyway, this is part of the question. So that ends. You can practice this before you take the exam on Friday. And the exam on Friday, I think hapon siya. Kalimut ko sa oras. 3 to 5 or 1. So I will just post. So just be ready by Friday, no? 12, dapat ready na inyong setup. Ready na mo. And if there are instances that you will be asked to upload the pictures, okay, of your structures. For example, you'll be asked to draw the structure. Let's say here in the problem set, draw the structure, the Fischer projection or the Haworth projection of your disaccharide.
Okay, so, or your monosaccharide, padro mo, and then wala mo, dili siya multiple choice. So, pag inana na you take picture of your answer, magsulat mo sa paper, you take picture of that, then i-upload ninyo. So, pwede nyo i-upload sa Google Drive or pwede nyo i-upload directly dito sa indicated slot sa question. So, either way. So, there will be case siguro nga inana. So, I don't know pa sa exam kung but the coverage is chapter 1 to chapter 3. Chapter 1, so you have to remember itong apela itong mga stereoisomers, no? Katong mga chiral compounds. Reviewhan ninyo na sa inyong organic chemistry katong chapter 1, balik ka to ninyo. Unsa nang mga D, glyceraldehyde, nganong D, monosaccharide atong gamit. Okay, unsa pa. So also your proteins, unsa tong mga amino acids, kato yung mga structures into itong mga polar or hydrophobic monosaccharides kadto. So kung gusto mo, pag-andam na lang mag-list daan sa inyong mga amino acids, and then even na list ninyo sa inyong monosaccharides. Pwede i-print ninyo. Print ninyo itong part sa PowerPoints, no? Sa pa inyong andaman. Pen and paper also, kanang blanco, bond paper kung pwede. Para limpyo. Then pag mag-take mag-exam, dapat pag mag-take mag-exam, dapat na may pen and paper para maka-jot down. No? And then also practice, we have a Google Drive. Pwede nyo i-upload dito ang inyong problem. And then kung link inyong ihatag, copy-paste. So make sure that you copy-paste anyone with the link, no? And then you paste it sa indicated cells. So either way, but I will suggest i-upload na lang nyo dito ang picture sa inyong nasulat dito sa indicated slot, mas dali ra kasi mo time. So either way, depends on you. What else? So chapter 1 to 3, if you have answered your problem sets, I think you can answer the midterm exam. Majority will be multiple choice, but there will be instances that you'll be asked to draw on the structure. So if that would happen, again, make sure that you have a clean paper or clean notebook now where you can take picture. I would highly appreciate if inyong magsulat-sulat ng structure walay linya. Kapag pangit mo tanawon kung linya-linya mo mo elementary and high school. No? So make sure that you have a bond paper. It's a clean or any clean paper. Walay linya, so that you can easily draw your structures. Okay, that's all our session for today. Do not forget to indicate your attendance in the LMS. If you have questions, you can ask questions.
Ma'am, good afternoon, ma'am.
Yes.
Ma'am, ganang available na ang attendance sa kwan, ma'am? LMS, ma'am?
Yes, it's already in the LMS.
Okay, ma'am. Thank you, ma'am.
Okay, if you want to review, answer questions, tanaw na nyo daan ng mga questions sa katong sige na to. Atong textbook na to, Introduction to Organic and Biochemistry nga book, no? And there is this chapter 12 and chapter 13, 14 dito. Chapter 12 and chapter 14 kani. Ito mo pag-review para if you want to be perfect, no, sa inyong exam. Kani. Kani, Introduction to Organic and Biochemistry. I think na-upload na ako. So, proteins is chapter 14 dito. And you can go to end questions. Answer ito ninyo. If you can answer katong sa last part, maka-answer mo. Nadirin ako ikuha yung problem sets. Much better. You can answer all those. And then chapter 12, dito sa end questions also. Kung gusto mo mag-study, who knows di ay no kung na yung gawas diri.